Hello everyone, welcome to the fourth module of the paper Cell Biology. In this module, we will study about the ultrastructure of the ribosomes and the process of protein synthesis. This module would help us to gain a pretty good idea about the structural and functional organization of ribosomes and the distinguishing features between prokaryotic and eukaryotic ribosomal protein synthesis. Structure and function of ribosomes. Eukaryotic cells are distinguished from prokaryotic cells by the presence of membrane enclosed organelles within their cytoplasm. These organelles provide discrete compartments in which specific cellular activities take place, and the resulting subdivision of the cytoplasm allows eukaryotic cells to function efficiently in spite of their large size. Ultrastructure of ribosome. Ribosomes are sites of protein synthesis in both prokaryotic and eukaryotic cells. Ribosomes are usually designated according to their rates of sedimentation, 70S for bacterial ribosomes and 80S for the somewhat larger ribosomes of eukaryotic cells. Both prokaryotic and eukaryotic ribosomes are composed of two distinct subunits, each containing characteristic proteins and ribosomal RNAs. The fact that cells typically contain many ribosomes reflects the central importance of protein synthesis in cell metabolism. E. coli, for example, contain about 20,000 ribosomes, which account for approximately 25% of the dry weight of the cell, and rapidly growing mammalian cells contain about 10 million ribosomes. The structure of prokaryotic and eukaryotic ribosomes is similar, although they differ in some details. The small subunit designated 30S of E. coli ribosomes consists of the 16S ribosomal RNA and 21 proteins. The large subunit 50S is composed of the 23S and 5S rRNAs and 34 proteins. Each ribosome contains one copy of the ribosomal RNAs and one copy of each of the ribosomal proteins, with one exception. One protein of the 50S subunit is present in 4 copies. The subunits of eukaryotic ribosomes are larger and contain more proteins than their prokaryotic counterparts have. The small subunit of eukaryotic ribosomes is composed of the 18S rRNA and approximately 30 proteins. The large subunit 60S contains the 28S, 5.8S, 5S rRNA and about 45 proteins. Like tRNAs, rRNAs form characteristic secondary structures by complementary base pairing. In association with ribosomal proteins, the rRNAs fold further into distinct three-dimensional structures. Functions of ribosomes. Ribosomes are the workhorses of protein synthesis, the process of translating mRNA into protein. The mRNA comprises a series of codons that dictate to the ribosome the sequence of the amino acids needed to make the protein. Using the mRNA as a template, the ribosome traverses each codon of the mRNA, pairing it with the appropriate amino acid provided by an amino acyl tRNA. Amino acyl tRNA contains a complementary anticodon on one end and the appropriate amino acid on the other. The small ribosomal subunit, typically bound to an amino acyl tRNA containing the amino acid methionine, binds to an AUG codon on the mRNA and recruits the large ribosomal subunit. The ribosome contains 3 RNA binding sites designated A, P and E. The A site binds an amino acyl tRNA, the P site binds a peptidyl tRNA and the E site binds a free tRNA before it exits the ribosome. Protein synthesis begins at a start codon AUG near the 5' end of the mRNA. mRNA binds to the P site of the ribosome first. The ribosome is able to identify the start codon by the use of the Shine-Delgarno sequence of the mRNA in prokaryotes and Kozak box in eukaryotes. Although catalysis of the peptide bond involves the C2 hydroxyl of RNA's P site, adenosine in a protein shuttle mechanism, other steps in protein synthesis are caused by changes in protein conformations. Since the catalytic core is made of RNA, ribosomes are classified as ribozymes, and it is thought that they might be the remnants of the RNA world. Both ribosomal subunits, small and large, assemble at the start codon. The ribosome uses tRNA that matches the current codon on the mRNA to append an amino acid to the polypeptide chain. This is done for each triplet on the mRNA, while the ribosome moves towards the 3' end of the mRNA. Usually in bacterial cells, several ribosomes are working parallel on a single mRNA, forming what is called a polyribosome or polysome. Ribosome, a protein factory. A ribosome is basically a very complicated but elegant micro machine for producing proteins. Each complete ribosome is constructed from two subunits. A eukaryotic ribosome is composed of nucleic acids and about 80 proteins and has a molecular mass of about 4,200,000 Daltons. About 2/3 of this mass is composed of ribosomal RNA and 1/3 of about 50 plus different ribosomal proteins. Ribosomes are found in prokaryotic and eukaryotic cells in mitochondria, chloroplasts and bacteria. Those found in prokaryotes are generally smaller than those in eukaryotes. Ribosomes in mitochondria and chloroplasts are similar in size to those in bacteria. There are about 10 billion protein molecules in a mammalian cell, and ribosomes produce most of them. A rapidly growing mammalian cell can contain about 10 million ribosomes. That is, a single cell of E. coli contains about 20,000 ribosomes, and this accounts for about 25% of the total cell mass. The proteins and nucleic acids that form the ribosome subunits are made in the nucleolus and exported through nuclear pores into the cytoplasm. The two subunits are unequal in size and exist in this state until required for use. The larger subunit is about twice as larger as a smaller one. In the ribosome, there are 3 stages and 3 operational sites involved in the protein production line. The 3 stages are 1. Initiation, 2. Elongation, 3. Termination. The 3 operational or binding sites are A, P and E, reading from the mRNA entry site. Sites A and P span both the ribosome subunits, with a larger part residing in the ribosome large subunit and a smaller part in the smaller subunit. Site E, the exit site, resides in the large ribosome subunit. The 3 stages. Initiation. During this stage, a small ribosome subunit links onto the start end of an mRNA strand. Initiated tRNA also enters a small subunit. This complex then joins onto a ribosome large subunit. At the beginning of the mRNA strand, there is a start translating message and a strand of tRNA charged with one specific amino acid enters the site A of the ribosome. Production of a polypeptide has now been initiated. For the tRNA not to be rejected, the 3-letter code group it carries which is also called an anticodon, must match up with the 3-letter code group called a codon on the strand of mRNA already in the ribosome. This is a very important part of the translation process, and it is surprising how few errors of translation occur. In general, the particular amino acid it carries is determined by the 3-letter anticodon it bears. Example, if the 3-letter code is CAG, that is cytosine, adenine, guanine, then it will select and transport the amino acid glutamine. Elongation. This term covers the period between initiation and termination, and it is during this time that the main part of the designated protein is made. The process consists of a series of cycles, the total number of which is determined by the mRNA. One of the main events during elongation is translocation. This is when the ribosome moves along the mRNA by one codon notch and starts a new cycle. During the startup process, the initiation tRNA will have moved to the site P, and the ribosome will have admitted into site A a new tRNA charged with one amino acid. The charged tRNA resides in site A until it has been checked and accepted or rejected, until the growing peptide chain attached to the tRNA in site P has been transferred across by enzymes to the charged tRNA in site A. Here one new amino acid is donated by the tRNA and added to the peptide chain. By this process, the peptide chain is increased in length by increments of one amino acid. The peptide bond formation between the growing peptide chain and the newly admitted amino acid is assisted by peptidyl transferase and takes place in the large ribosome subunit. The reaction occurs between tRNA that carries the nascent peptide chain, peptidyl tRNA, and the tRNA that carries the incoming amino acid, the aminoacyl tRNA. When this has taken place, the tRNA in site P, having transferred its peptide chain and now without any attachments, is moved to site A, the exit site. Next, the tRNA in site A, complete with the peptide chain increased in length by one amino acid, moves to site P. In site P, riboproteins act to consolidate the bonding of the peptide chain to the newly added amino acid. If the peptide chain is long, the oldest part will be moved into the cytoplasm to be followed by the rest of the chain as it is produced. The next cycle with site A now empty, translocation takes place. The ribosome moves on by a distance of one codon notch along the mRNA to bring a new codon into the processing area. tRNA charged with an attached amino acid now enters site A, and provided a satisfactory match of the mRNA codon and tRNA anticodon is made, the cycle starts again. This process continues until a termination stage is reached. Termination. When the ribosome reaches the end of the mRNA strand, a terminal or end of protein code message is flagged up. This registers the end of production for the particular protein coded for by this strand of mRNA. Release factor chemicals prevent any more amino acid additions, and the new protein is completely moved out into the cytoplasm through a cleft in the large subunit. The two ribosome subunits disengage, separate, and are reused or broken down. Let us now summarize what we have learnt in this session. Ribosomes are found free in the cell cytoplasm and also attached to the rough endoplasmic reticulum. Ribosomes receive information from the cell nucleus and construction materials from the cytoplasm. Ribosomes translate information encoded in the messenger ribonucleic acid mRNA. They link together specific amino acids to form polypeptides, and they export these to the cytoplasm. Each ribosome is a complex of ribonucleoproteins with 2/3 of its mass composed of ribosomal RNA and about 1/3 ribosomal protein. Protein production takes place in 3 stages: initiation, elongation and termination. Here are a few questions for you to work out. 1. What is the role of ribosomes on endoplasmic reticulum? 2. What is the role of rough endoplasmic reticulum in protein synthesis and post-translational modification of proteins? 3. How are proteins targeted to the endoplasmic reticulum? 4. Explain the structural organization of the Golgi complex. 5. Ribosome is commonly called as a protein factory. Why? Here are a few books for your reference. The Cell by Jeffrey M. Cooper, Robert E. Hausman, A Molecular Approach, 3rd edition, ASM Press. Molecular Biology of the Cell by Bruce Alberts, Alexander Johnson, Julian Lewis, Martin Raff, Keith Roberts and Peter Walter, 4th edition, New York, Garland Science, 2002. Biochemistry by Jeremy M. Berg, John L. Timosco and Lubert Stryer, 5th edition, W. H. Freeman, 2002. Here are a few websites for your reference: www.ncbi.nlm.nih.gov/books/nbk9930, www.bscb.org/softcell/ribo. Thank you for watching this session. Let's meet in the next session with another topic. Till then, bye.